All right, guys, so now let's go ahead and focus on eukaryotic cells. We're going to focus on eukaryotic cells. Eukaryotes. All right. And we already mentioned that one characteristic of eukaryotic cells is that those are going to be cells that have a nucleus, right? So let's go ahead and get started because you know, guys, I like drawing my stuff, yeah. And we're going to start putting all the pieces together and all the organelles and everything that takes a role in the making and the function and keeping that cell working effectively. And number one, if you guys remember, this is where the lipids take place. We have this phospholipid bilayer made out of free fatty acids right here in the middle with, there is going to be non-polar right there in that section. So we have our phospholipids. It's going to be free fatty acids. So this section of the plasma is going to be non-polar. Therefore, it's going to be hydrophobic, right? Hydrophobic. So we know that. And right here in the section of the other one is going to be made out of a glycerol molecule with a phosphate group and a choline molecule. That's going to make it polar, right? Which is going to be hydrophilic. So if you guys remember from the chapter last time that we're talking about osmosis, diffusion, and everything, this is very important because outside of the cell, we're going to keep the plasma, we're going to keep it in contact with water, and inside of the cell, we're going to have all that cytoplasm that is going to be mostly about water. So it's very important to have that phospholipid bilayer right there as part of the plasma membrane to make sure that we keep that a selective barrier, and this barrier is able to keep in check of what goes inside of the cell and what goes outside of the cell. So remember that we have that plasma membrane, right, made of phospholipids, okay? So we have that to begin with. Also remember that we were discussing that in prokaryotic cells, they have that extra thick cell wall in some of them that confers that extra layer of protection and of resistance. But we don't have that in eukaryotic cells. We just have that very thick plasma membrane, just keeping track of everything that goes inside and outside of the cell. So we discussed that we have a nucleus, right? So it's very important that we have a nucleus. And if we're saying that the plasma membrane is a selective barrier for molecules, solutes, ions, water, anything that goes inside and outside of the cell, the nucleus is going to be more selective, way more. So in order to go inside, nuclear envelope. So in order to pretty much go inside of the nucleus, you have to just have a VIP card, right? You cannot just enter the nucleus like the plasma membrane. It's just going to be highly selective to a very particular components regarding genetic information. And that's it. If you don't have that plasmine card that allows you to cross that nuclear pore complex, just forget about it. You will not be able to either exit or enter that nucleus. So talk about the nucleus. That nucleus is going to have an extra nuclear envelope protecting all the genetic information. We know that inside of that information we have the DNA, right? So that's going to be the holy grail. It's going to just keep it like in a very secured, safe right there in the nucleus. So we have the DNA, and it's going to be protected by that nuclear envelope. And that nuclear envelope is just going to have this very thin layer inside of it. And that very thin layer, right, is going to be able, made out of plasmine, right, out of plasmine. And that layer is pretty much going to be able to give that extra layer of protection to make sure to maintain the integrity of the nucleus, like right in place. Now, inside of the nucleus, we're going to have another important organelle. And that organelle is just going to be in another smaller right there. And that is going to be our nucleolus, right? So that's going to be the nucleolus. Just a little difference right there. So that's going to be the nucleolus. So one very special thing happens in the nucleolus, right? And that is going to be the assembly of the ribosomal RNA. So only the ribosomal RNA that is important for the assembly of making ribosomes, right? And the ribosomes, we're going to be able to find them outside in the cytosol. They're important for the making of proteins, right? Coding all the information to make the first translation into amino acids. So that information of the ribosomal RNA is pretty much assembled right there inside of the nucleus, right? So we're talking that we have the nucleus. The nucleus, we know it holds DNA. The transcription process happens inside of the nucleus, right? So all the RNA, the DNA that is transformed and transcribed into RNA happens inside of the nucleus. Just the only thing that is going to happen inside of the nucleolus is going to be the assembly of the ribosomal RNA. So far we have the DNA, we have the RNA, but RNA cannot exit the nucleus as RNA. You cannot learn later on in other chapters how this process is going to be taking place and maturing into messenger RNA, but RNA has to be evolved and matured into messenger RNA in order to be able to exit the nucleus as messenger RNA. So remember that RNA by itself cannot exit the nucleus. It has to be processed and matured as a messenger RNA in order to exit the nucleus. So the only guys that we have so far that will be able to exit the nucleus is going to be our ribosomal RNA and our messenger RNA, okay? And for the ribosomal RNA and the messenger RNA, in order to exit the nucleus, they're going to have to walk through what we call the nuclear pore complex. So the nuclear pore complex is just going to be some channels that we have right there. Just imagine these nuclear pores. There's going to be pores, but there's going to be a protein right there. Just imagine they're like little law enforcers that they have to have a specific card to detect and be activated, right, in order to open and allow the messenger RNA or the ribosomal RNA to exit that nuclear pore complex and go into the cytosol. Now, we're talking that those are the only ones that are able to exit the nucleus. There's going to be also some substances like proteins that have kind of like a zip code with a shipping address that says nucleoplasmin will have the ability to enter that nuclear pore complex and make it into the nucleus. Those proteins are going to be they're going to be necessary because we require some proteins and some molecules for the assembly of DNA, right? So those are going to be they're going to be very specific in order to pass that nuclear pore complex. Okay, so far we have the nucleus, we have the nucleolus, we have the nuclear pore complex, right? All of these systems under the nuclear envelope. Now we're going to go ahead and talk and follow the sequence of the genetic information. So we know that the main point of having DNA is that DNA has many thousands of genes, and all of these genes are pretty much so we can produce proteins, right? So we're going to follow that train of thought in the making of the protein. So we have the nucleus with the DNA, transcription process in the RNA, and now we want to be able to exit that messenger RNA and be able to make a protein. So the first place where that messenger RNA is going to be these little places, right? I'm just going to put like a little dots in there. And these guys are called ribosomes. I'm going to put many of them. But imagine these ribosomes. They look like little mushrooms. I'm just going to put a mushroom right there. Imagine kind of like that picture. You need a little bit of imagination. So we're going to put the large subunit and the small subunit. We were mentioning in the previous video of making the differences between prokaryotic and eukaryotic cells is that they are composed of ribosomes of large subunits and the small one. And the only differences between ribosomes between prokaryotic cells and eukaryotic cells is just going to be pretty much the size of those subunits, and that's it. Also, let's remember that these ribosomes are assembled by the making of that ribosomal RNA that is coming from your nucleolus, right? So we need that ribosomal RNA that is going to take place in those subunits. So, okay, so we have the ribosome. Ribosomes are not considered organelles, okay, because they do not have a membrane. So that's the only characteristic that separates them from label them as an organelle. So they're going to be these macromolecule. These are very complex macromolecule machines, and their ability is just going to rely that the messenger RNA is going to exit the nucleus, it's going to drive all the way to a ribosome. We have many of them, so we are going to have that messenger RNA. So just imagine that we have a sequence of RNA, UUU, UAA, and so on. This is a stop codon, but anyways, just to quickly put a UUU and a UAA to make it like a stop codon really fast. So this sequence of information is going to come inside of the ribosome, and ribosomes are going to be in charge of decoding all of that genetic information. They have to pretty much every codon that they have converted into amino acids, right? So we have a peptide bond, amino acid, a peptide bond, and that, another amino acid right there. Just a quick example, like a big representation. So this decoding of information and translating the information, that's why it's called translation, because it's going to translate all these messenger RNA and to make the primary structure of a protein. It's not a fully functional protein yet, but we have a first the primary structure of a protein right here. And let's remember from the chapter of proteins that proteins are made of these peptides, right? Amino acids connected with a peptide bond, amino acid, peptide bond, amino acid. Very simple like that, just the sequence of amino acids lined up together, right? So that is going to be happening right there on the ribosomes. So we have many ribosomes, right, because the information we produce in many proteins and information like per second very quickly. So this is happening really fast. Now, in order for that primary sequence of amino acid to be converted into a fully functional protein, this information needs to be deposited, needs to be sent out to another organelle. Now, we're going to have two organelles that their names are very similar, but they're going to have a completely different function and name, right? So that is going to be the rough endoplasmic reticulum. So this rough endoplasmic reticulum, you're going to see, and then we have it on one side, and on the other side, I'm just going to put the smooth endoplasmic reticulum. Okay, so right here we have the rough endoplasmic reticulum and the smooth endoplasmic reticulum right there. And okay, so we have the genetic information right here coming out, and we say we now need to label it and make it a fully functional protein. So just imagine that you order something from Amazon, right? You want something to be able to be sent to your home address. So right here in the factory, the rough endoplasmic reticulum, let's just say the Amazon warehouse, right? They're going to make sure to get that information, get that order that they just got, and these guys going to come inside here. The amino acids is going to come inside here, the rough endoplasmic reticulum, and it's going to be delivered inside. Now, inside of the rough endoplasmic reticulum, all this information is going to start be packaging, folded, package it and put it in a nice little structure in a box with information that can be later on be sent to your home address, right? But for now, we need to put some bubble wrap, we need to fold it, and we need to put that structure and form and shape to that protein. So it's going to come inside the rough endoplasmic reticulum, and all of those hydrogen bonds of the secondary structure are going to happen inside of there. All of the tertiary structure, right, of all the different type of bonds that we have, the polar, nonpolar, acidic, basic, covalent, and all of these type of bonds, they're going to happen right there at the rough endoplasmic reticulum. So they're going to exit right here as a 3D protein right here by the end. So the rough endoplasmic reticulum, what you can remember is that is pretty much involved in the folding of the primary sequence of the amino acid, all the different forms, all the alpha, beta helices and the beta pleated sheets are going to be formed right there in the rough endoplasmic reticulum. all the extra bonds that are needed to create a 3D molecule are going to happen right there. And there's going to be another important component, if you guys remember from the previous chapter on proteins, there's going to be right there in some molecular chaperones. They're called like that. They're like little supervisors of the warehouse. They're making sure that all of this folding process from the beginning until the end is done properly. Remember that proteins need to have a specific structure and a specific folding in order to work effectively as a protein. If the protein is not folded correctly, right, it's going to be completely an erroneous protein. It's not going to function properly, and it can give us even a disease or a problem, like sickle cell disease, right? If the information is not coded right, interpreted right, folded right? Instead of having like a good red blood cell, we're going to end up with a sickle cell blood cell that is not going to function effectively when higher demands of oxygen are needed. Okay, so that's for that. So we have the 3D protein right there. So still, yes, it is folded. It's a 3D protein, a tertiary structure. We have the secondary structure of a protein right here, a secondary. We have the tertiary structure, which is going to be the 3D protein, right? Right there coming out. Now, if you notice something here, I put like a little bubble like around it. It has to come out when the process exits the rough endoplasmic reticulum. This protein is going to steal a little of that envelope, that membrane of the rough endoplasmic reticulum. Remember that our organelles have a membrane, right? That's why they're called organelles, and that's why our ribosomes are not organelles, because they do not have a membrane. So when the protein exits that process, it's going to exit through a process that we call exocytosis. So this protein is going to steal a little bit of that protein. It's going to wrap itself around. It's going to cover itself so it can go ahead and be delivered to the next organelle, right? So that's going to be very important to understand. Now, after we have that vesicle made out of protein, it already is in a 3D shape. This protein needs to have a label. It needs to have a shipping address, right? So you can make it to your home, to the order that you made initially from the nucleus, right? So you need to have that package delivered to your home. Now, proteins can take different roles. Proteins can stay inside of the cell and assist with a lot of these complex interactions and activities that are happening. Proteins can also go ahead and take a place of a peptidoglycan, a protein, a channel, a pump, something that is part of the plasma membrane, so it can be sent there. Or another thing is that we're going to ship it. Something is going to stay local, it's going to go somewhere international, and we're going to send that protein somewhere else. So these proteins need a label. It's a shipping address, right? It needs that zip code. So for that, we're going to send that protein into the Golgi apparatus. It's going to be right here. We have that Golgi apparatus. Sorry, everything looks the same. Apologize for that. So we're going to put Golgi apparatus. What colors am I using? Oh, this should be rough endoplasmic reticulum. Rough endoplasmic reticulum. We're going to talk about the smooth endoplasmic reticulum in just a bit. So we have the rough endoplasmic reticulum, and we're going to have here the Golgi apparatus. Right? And that Golgi apparatus is going to make sure that this protein, this vesicle that contains a protein, enters here, right, our Golgi apparatus. And as that protein enters the Golgi apparatus, it's going to get rid of that membrane that is stored from the rough endoplasmic reticulum, remember? So it's going to get rid of it. And that process is going to be a endocytosis process. So as it exits, it's going to be exocytosis, and as it leaves the membrane and gets rid of it and enters a new organelle, that's going to be endocytosis. So this protein, I'm just going to put protein, a fibro protein is going to come inside. Many things can happen in the Golgi apparatus. Golgi apparatus, yes, it's going to be a label, it's going to give a molecular tag to it, it's going to put like a print to it, right? kind of like a little tag. So that protein is going to exit with a little tag. And that tag is just going to have that molecular zip code. And that zip code is going to tell the protein if it's meant to stay inside of the cell and assist the locals, if it's meant to go to the plasma membrane, or if it's meant to go international and just take some higher role outside of the cell. But another thing can happen as well. Sometimes these proteins are glycosylated. Glycosylated simply means that they're going to add some sugar on top of it. I'm going to put some extra here. That's supposed to be sugar, okay? You just imagine that I'm adding some sprinkles to it, to that protein, right? So that protein is going to have like some sprinkles. It's going to become glycosylated, right? That means it has some sugar, right? And sometimes that protein can become like part of the membrane, right? We know that a lot of these protein carriers and protein channels, they're located right here, taking a role. And they're mostly going to be like glycoproteins or glycolipids, right? Or different types of combinations that assist in the passage and being kind of like law enforcers of the plasma membrane, right? Or very specific, let's just say that this protein is going to be in charge of carrier molecules of glucose and assisting them to come inside of it. Remember that also, like channels are very specific to what substrates they're connected with. So we have that glycosylation, we have that molecular tag, and sometimes this one can also be sent to other organelles. So we have there that now this protein is ready to go. It's already a fully proper functioning protein. It has a shape, it has a function, it has a molecular tag. It's good to go, right? To go and do its role. So we have the rough endoplasmic reticulum, we have the Golgi apparatus, right? And then we have the smooth endoplasmic reticulum. So right here on the smooth endoplasmic reticulum, even though they're kind of like very similar, there's going to be one main difference if you already noticed, right? And that's going to be the presence of ribosomes. So rough endoplasmic reticulum, when you look at them in the microscope, you can make the difference by knowing that these ones are going to be all surrounded. They're surrounded completely by the ribosomes, because ribosomes are just receiving orders all day long. People want their stuff, they want their proteins, right? So they're always receiving orders, they're always translating information, they're decoding information, and they're always like making sure that they're delivered to the rough endoplasmic reticulum, and they're delivering those proteins really fast, right? So that's going to be the characteristic of this one. And in the smooth endoplasmic reticulum, we don't have the presence of ribosomes. In the smooth endoplasmic reticulum, what happens, we have is the synthesis of lipids. So like the proteins are here for the rough endoplasmic reticulum. The smooth endoplasmic reticulum is going to be in charge of the synthesis of lipids, okay? There's also going to be an important storage room for calcium ions. So smooth endoplasmic reticulum are in charge of storing calcium. All that calcium, deposits of calcium, they store them there, and they're sure that they keep them in case the cell needs it for many different things, right? Okay, so we already have those organelles. Now we start adding more puzzles to these more pieces to the puzzle, a little bit the opposite. So we're going to have here, I'm going to put a lysozyme. That's going to be a lysozyme. I'm just going to put an L right there for lysozyme. Now, lysozymes are very important because lysozymes are like a center, a recycling center. So let's just say that we have a protein. A protein is not working very well. It's an error. We don't like it. It's malfunctioning. You know what? Let's get rid of it. So that protein might come to the lysosome center. It's going to be, the lysosome is going to release a lot of these acid hydrolases, right? They're going to release that. They have like a pH of around 5 about. So they're going to release all of these acid, and they're going to go ahead and disintegrate it can be like different molecules. It can be a protein, a carb, something else, a different organelle, something that is not working. So they go ahead and disintegrate it, and sometimes it's engulfing, like, or recycle some of those materials from those macromolecules for something else, right? So there you go. Those are going to be your lysosomes. Sometimes proteins, they're going to be assisting in the making these hydrolyses, be part of it. So because the rough endoplasmic reticulum, the Golgi apparatus, and the lysosomes work together, they just got together. They created their own system. They created a band, like I always say, right? And they call it the endomembrane system. So all of those guys, remember, RER, Golgi apparatus, and the lysosomes. They have their own band that is called their indie band, right? That is called the endomembrane system. They sound like an indie band to me. All right, so we have that. Let's start talking a little bit more. We're also going to add here peroxisomes, right? I'm going to put a P, I'm sorry, just to make it simple. I know it's the green. We have the peroxisomes. So the peroxisomes are going to be in charge of doing a lot of the redox reactions. Redox reactions happen right there. Oh, it doesn't want to paint, but it's going to be an R right there. So all the redox reactions happen right here, and they assist with that, right? So peroxisomes is like a center where a lot of enzymes take place, redox reactions take place, and these redox reactions and the release of that enzymes, they're going to assist in other catabolic reactions that are going to happen in other organelles as well. So pretty much we're going to spend the next chapter on enzymes talking about a redox. So just keep that in your mind, okay? We're going to put a little bit of hold into it until the next chapter. Just have that like redox information in your mind for the next chapter. Now we have another one, and you know, guys, that's always my favorite, right? And those are going to be that's going to be our mitochondria right there, right? That's my favorite. Those are my guys. I was going to put ATP, but it's going to be mitochondria. The mitochondria, right? And that's going to be the powerhouse of the cell, right? Mitochondria are going to be in charge of breaking away macromolecules. So let's just imagine that we have a molecule of glucose coming up our way because we need energy. We have an exos right here, right? I'm going to put a glucose. It's going to come inside. This glucose is going to be broken down through a process that is called glycolysis. Glycolysis is going to be separated. It's going to give us two molecules. It's still going to be simplified into other molecules. It's going to go under different reactions, and then it's going to enter a process that is called cellular respiration right here in our mitochondria, right? And that is very important because it's going to create all of these macromolecules and start to produce all of these ATP, right? So we pretty much, every cell, we require about 10 million of all of these ATPs like per second. So cells are always for different reactions that are happening for movement, for enzymatic reactions, for the production of a lot of this stuff and part of the pumps. So the cell requires a lot of ATP for energy and be able to create and produce that energy and to keep, the function and things working neatly and effective. Now, the more you're going to see later on that your body has more energy that you require or more oxygen that you require. Let's just say that I'm working out and I need like a lot of ATP. My body is going to require more ATP in the process. So you're going to be, when you're running, you start breathing faster, right? Because your mitochondria is requiring all of that oxygen to produce ATP. That's pretty much kind of like over 90% of the reason that we breathe. We require that oxygen because it's going to go into your mitochondria to help us produce that ATP, right? It's a very complex and very beautiful system, I think, right? So we have that ATP, and the more that you work out, you're going to see that it's going to increase your mitochondrial density. That means that more mitochondria are going to start appearing in your cells because the demands are high and your cell is requiring more ATP than you normally do until normal and rest conditions, right? So your mitochondria can multiply, can increase. You can see that it's recycled. Mitochondria is about every 10 days. So that's why you're going to see that it takes about 2 weeks that you start, if you stop working out, yeah, you kind of like, I started losing my fitness levels. Yeah, because mitochondria recycle about every 10 days, right? And we're recycling those mitochondria. All right, very cool. So those are going to be the ones that are in charge of giving us all of that ATP. And what other thing am I missing, guys? We have all the system created together. I'm trying to think what am I organelle I might be missing. So I think I'm going to leave it there. But yeah. Okay, so one more thing. Those are going to be the primary organelles. Make sure to check them. The cytosol is also, the cytosol is going to be, just imagine kind of like the water, everything that is part of the cell. Remember that in your cell is made up 60-70% of water, so all the water is going to be inside of your cell. So, but also the cell has a cytoskeleton. Like in our bones, like in our body, we have our bones that give us shape and structure and help us to be solid in our bodies and strong, right? Also, the cells have different types of fibers that help us to maintain the integrity and the shape of the cell. Right now, we have this beautiful circle cell pointing out the basic elements of a cell, but you can see that in our cells, they have different shapes and sizes. Some of them are going to be long, like the muscle fibers. They're just all of these long and elongated muscle fibers, right? And they're going to be consistent of different type of proteins and filaments. So all of the cytoskeleton is made up of different type of filaments. So for the type of filaments, we're going to have, number one, we have that tubulin, right? Tubulins, and we have actins, and let me release that tubulins. I'm going to put actins and myosins. We have actins and myosins. For example, these are going to be very important in your skeletal muscle fibers, right? So actins are just going to be little beads that are connected, kind of like that. I'm going to put a different color right here. They're just intertwined between them. But in between that, they have like this storage site. When they're activated, right, by the presence of calcium, what's going to happen that this is going to go ahead and twist around, open up a space, so they can connect with another type of myosines that are called these myosin fibers. These are another type of tubulin fibers, right? But they attach to it, and they are able to pull each other and make a contraction happen. So those are type of tubulins, not type of tubulins, I'm sorry, actin and myosins. So those are going to be your actins, right? I'm thinking of tubulins, but no, these are actins. And these pretty much help the muscle contract and pretty much like that. So for example, in the case of your muscle fibers, they're now like all pretty circular like that. These are going to be elongated fibers, right, that assist us with muscle contraction. Another part of the cell cytoskeleton is going to be your intermediate filaments, right? Filaments. And this is an example is going to be keratin. Keratin is pretty much provide support to some of the cells. You can find them in your nails, like in your hair, right? So they're a very important part of that structure in your cells. And now we have the tubulins, finally, because I'm here talking about tubulins. And these tubulins are also going to be important. We have some important, for example, centrioles. These centrioles help us. You're going to find them here and there. When we're talking about the cell cycle, you can remember those guys. Right now, they're just steady. They're being part of that structure. But when we need to create a new copy, an exact same replica of the cell, these guys are going to help us to break apart everything, separate everything, and now that's when they're going to become active. So for now, just remember them. They're right there, like in peace. They're not doing anything for the moment. But those are going to be your centrioles. They're going to be important in cell cycle and in mitosis. So we have the tubulins. Tubulins are also part of some, for example, flagella and sperm cells, right? So we know the sperm cells have this flagellum that allows it to have that whip-like movement and move around. But if we go ahead and pretty much cut it and make like a horizontal cut of that sperm cell, I'm just going to pull it like that and make a cut right there, we're going to see, I'm going to put a circle right there. It's called an axoneme. And this axoneme is made of 9+2 system that pretty much we have these pairs of tubulins together. There should be 9 pairs, okay? Just imagine 9 pairs right there. And then 2 in the middle. And right there we have some DNA tubes right there connecting these pairs in between. So what happens is that when they're activated by the presence of ATP, they're just going to be kind of like moving back and forth, kind of like dancing, making like a movement like that, steady movement like that. And that is what's going to allow for this flagellum to be moving in a whip-like fashion by the shaking back and forth of those tubulins in that axoneme, right, in movement. So I think that's it for the eukaryotic cells, the organelles, important organelles, and also part of the cytoskeleton, some of those components. All right, guys, so make sure to review the information. This is a very simple, fun chapter. I love it. But if you have any questions, please let me know. Take care.